A very good evening to all of you. And myself is Asif Naqvi, and I'm the director and CEO of BDG Lifesciences. Previously, the company was known as Biodiscovery Group. And since we've changed it to BDG Lifesciences OPC Private Limited, we have not, we've stuck to our roots of group. Now, group is something very important. I would just like to brief you over here that why we started this organization and what was the reason, what was the motive of starting this organization. Now, as you can see, that group is there in the organization, the name of the organization itself, which means that it is for everyone. Now, when we started this organization in 2009, I wanted to help students. I I have recently completed my projects in Switzerland, and then I returned to India. And I was like that. I used to get so many queries with the students, especially at bachelor's and master's level, and they were not sure that what they had to do next after completing their bachelor's and master's. So the biggest thing which we analyzed at that time back in 2009, that the people, they don't have a strong research profile. People, they do not had anything in what we call in terms of research. And as we know that biotechnology or bioinformatics is it's research. It's a field which has a lot of research in it. And if you do not have any research papers or a good publications under your belt, then your life becomes very hard to achieve a good, I should say, a good job or a good PhD position. So, and then definitely there were some people, there was an information gap. And the information gap was that by the time the opportunity used to reach the talented person, the opportunity used to, it just used to vanish. So, the opportunity goes off. So there was this information gap between the talent as well as the opportunity. So we decided to form a group where we would help people, especially students and young professionals, and give them something which they are not getting anywhere. And back then in 2009, there was only one organization, Biotechnica was there, and they were doing a really good job at that time. And then today also, they are also doing really great. So we thought that why not to create a group and pass on the information. So that is how Biodiscovery Group came into existence. So I'll just quickly go through 10-15 minutes of yours, and I will just take this much time in telling about my company which I have started back then. And then I will hand over the session to Dr. Nagagireesh. He is also here. And I will not take much time of yours, so just I'll quickly proceed. So basically, who we are, what was the reason? I have already told you. As a group, the idea was to help. And then this is who we are, who we have become. And since 10 years of the organization, this is what we have been doing. We are a guide who will help you in solving your academic problems. Now, this is a very big sentence, but that is what we have been doing since the past 10 years, showing you the right path, solving your queries, giving you correct training, paving your path for a successful career, and giving you launchpad to excel into the field of science. So these are 6 points, or I should say 5 points, which are very important for every student who is in their final sessions of their degree, whether it is a BTech or whether it is an MTech or BSc or MSc. Now, back then, I have already told you that it was in the form of a Google group, but now Google groups are not there, and then Yahoo groups are also not there. So we have transformed into Facebook, Instagram, LinkedIn. So I would like to show you also that how many people are there, and then on which platform, and then you can subscribe to that. And all these services we are doing it for free, because that is what who we are, and that is what the organization, when we started this organization, that was the motive. So as I have already told you that biodiscovery group, the group itself means that we want to take everyone together and proceed. We want everyone, and then people from all over the world have joined us, and they have different kind of queries, they have different set of questions. However, the most important one is that, sir, what to do next, what we should do next, so that we can have a much better job opportunity, so that we can have a nice PhD opportunity. So all I and my team tell the students, like we have a 24-hour team that works in shifts on social media, that help and solves these queries for the students and research scholars. The basic point which we say, and which obviously I would like to mention it over here, that if you want to excel into the field of biotechnology or bioinformatics, then definitely you should have to have good publications under your belt. This is the only way of success in our field. Until unless you do not have good publications under your belt, good journals, good conferences, then life would be very hard for you. So people who are saying no jobs, there are no jobs, there are no jobs, first you need to understand, or you need to look into yourself, that how many research papers you have published. Now people will say that in 4-year degree course, how can we publish papers, right? But no, every curriculum, every university in India as well as abroad, they have major projects. In the last year, 6-month project, dissertation project is there. Try to work on a novel research project and get it published. That is your key to the next door, which will open much better opportunities. So this is what we suggest, and this is how we are running this organization, by helping people. So there are a number of things which people ask, which students ask, but then this is the basic one, and this is the most important one. So as I have already told you, in 2009, we created a group for students, researchers, scientists, and professionals who are related to the field of biotechnology, bioinformatics, and pharma. And this group has a free consultation, tutorials, project reports, and mainly job updates, as I have already told you, in India and abroad, then PhD scholarships, postdoctoral scholarships, and most importantly, latest science and technology news. Because I would like to point out one more thing over here, that we are from a field of biotechnology. And every new day, every single day, a new technology is coming up. So until unless we do not pace up with the technology, we won't be able to survive. For example, if you go for an interview and then a person asks you some discovery, like for example, who won the latest Nobel Prize for chemistry? Now, obviously that the two scientists who discovered gene and genome editing using CRISPR-Cas9, the great Jennifer Doudna and Ms. Charpentier, they have won it. So that is how you need to get updated, means update yourself with the latest science and technology news. That will give you an edge. So as of now, as an achievement of 10 years, we have more than 1 million subscribers, and these subscribers are not only students, faculties, lecturers, scientists, and company professionals, whom we help, whom we send our news letters. So that is a life, that is really great achievement for us. And now the next slide is over here. You could see that how many, at which place we have what kind of people. So these are the subscribers. You can see on social media, we've just put it like Facebook, we have 40%, then LinkedIn, then YouTube subscribers, and then Twitter followers. And then on messengers, like we have 2 platforms for WhatsApp, then What I Am, Google Duo, and Telegram. And then others, you could see Pinterest, Flickr followers, and We have our own page on our website where you can subscribe, where you can type the, where you can give your information, and definitely we can, we will, you can get subscription for our newsletter, and all of these services, it's free. So now let's come to the company. BDG Lifesciences Private Limited, formerly known as Biodiscovery Group. We are an expert in bioinformatics, and we provide services to the biotechnology, bioinformatics, and pharmaceutical companies. So basically, we are a contract research organization which does, for example, molecular docking, virtual screening, ADMET, and next generation sequencing analysis, and all these kind of bioinformatics stuff for our clients and companies. We started our operations in 2010, and these are our wide array of services like contract research, workshops, e-workshops, research projects, training, webinars, seminars, and conferences. Then you could see that this is the bar for our work of the business which we are doing. Research projects, you could see, because these research projects are online, so that people from all over the world, they are participating. Then workshops, e-workshops, which are online workshops, and then courses. Previously we used to do at least 2 courses. I'm talking about courses, free courses. So now I would like to introduce Envision with BDG initiative from here itself. So we used to do 2 to 3 courses previously before the COVID-19 pandemic. But then, since February 2020, we have increased our frequency. And as of now, we have done more than 70 courses in which more than 14,000 participants from all over the world have participated. So that is a big achievement. And as I have already told you that we used to do it previously, so we are not new to online mode of training and workshops, because we are doing online workshops since 2012. So this is what another thing. And then we have others like contract research. So contract research and project consultancies and research project consultancies. So this is what we have been doing. So our mission is to provide solutions for the future by catalyzing the process of biomedical sciences into therapeutic technologies that benefit human health and society, and eventually fuel the economic growth. So this is our mission. And as an organization, we continue to work into the direction of growth and expansion with an aim to serve more customers and make a difference to the society with innovative research work, employing latest technologies and excellent services. So this is what we have been, doing since 10 years. And we are thankful to God and thankful to all our participants that, they have liked our way of teaching, they have liked our programs. So this is one thing. You could see over here that these are the topics on which we are giving our services. You could see a number of topics are there, bioinformatics, and then in fact there are 2 workshops right now they are running. One is on bioinformatics, which is for beginners, and then molecular docking. So these 2 workshops are currently running. And then I would like to point out over here immune RepSec, that is immune repertoire sequencing. This is which we started in December 2019. And artificial intelligence and machine learning and biology, this we started in March 2019. So these are some very new programs which is attracting a lot of people from life sciences background. And then we have cancer, HIV, diabetes, and tuberculosis research, in which we are doing workshops, training programs, as well as research projects targeted, or I should say based on cancer, HIV, diabetes, and tuberculosis research. Quickly following to the next slide, this is value proposition. Now I would like to point out 4 terms in this: innovation, ideas, service, and value. Now this is very important Because innovation and creativity is the integral part of biodiscovery group or BDG Life Sciences. Why it has been this? Because we kept on introducing new ways of or new methods of teaching, as well as we used to put trainings and workshops on topics which are very important, which is the need of the hour, and used to keep on doing it successfully. And that is why innovation is to translate ideas into service that creates value. So basically innovative ideas we are translating into ideas that into service that creates value. And people who are getting training or getting the workshops, because these are all our hands-on workshops, so they are getting learning in a much better way, and that is definitely improving their skills, and definitely it is helping and strengthening their CV. So why I am stressing out on this slide, that is value proposition. Now value proposition means that why you need to be with us, why you need to come and do programs with us. Because practical application of technology, learn by doing the things. We learn by the things, and that is why since 2012 when we started doing the workshops, all our workshops are hands on. You need to perform on your own computer system, and that is how we have been doing many workshops around the world, and on site as well as online. Second, hands on training sessions. Whatever training which we conduct, all are hands on. Then novel research projects. Novel research projects, because if there would be no novelty in the research project, then obviously the person would be wasting time. So that is why novel research project is very important. Then result driven research. First novel research project, then result driven. So your research has to be result driven. And then lastly, everlasting relationship. Everlasting relationship is that we will keep on helping you until unless you do not want our help. So this is what this is why these are the 5 points by which is giving proof, and which is the concrete of BDG Life Sciences, which you guys should look up and decide that yes, I want to do programs at BDG Life Sciences. I hope you can see over here. I would not stress us on this slide, only just pass by with this slide by saying that this graph is going up. Now, what we started in 2010, if we would have not been doing anything new, then this would graph at one point of time would have came down and then saturated and then ultimately would have gone down. But then this graph is going up. This means that we are doing innovative and creativity work, and that's why that is going up. So just to summarize, we are a 10 years old company, and 60 plus research projects we have done as of now. And these projects are being done by people who are in their BTech or MTech or BSc or MSc, or faculties and company professionals who want to strengthen their profile by getting good publications. So, and then 400 plus workshops, that is on-site workshops in India, and then 20 plus workshops in other countries like Mexico, Sudan, Nigeria, Kenya, Egypt, Dubai, Thailand, and Malaysia. So these are the countries in which we have worked. However, in 2020, we had workshops in Amsterdam, in Prague, and in Singapore. But then we had to cancel it because of the COVID-19 pandemic. In fact, we have not canceled it, we have postponed it. Then we have been working at premier institutes. A lot of workshops we have been doing regularly at, for example, AIMS, All India Institute of Medical Sciences, New Delhi, IIT Guwahati, NIT, VIT, that is Vellore Institute of Technology, MMC, Madras Medical College, and then JIPMER, etc. And then lastly, happy customers. Now, happy customers are something very important, because if you could see on our YouTube channel, we have started posting videos of our feedback, which is very important, because a person who has never been to BDG Life Sciences or have never heard about BDG Life Sciences, at least they can look to the video and see that what was the experience of the person who has already done the program. So we have started uploading the video feedbacks. Previously it was the written feedback, then people used to, then the next phase was people used to post it on our website, and then now we have the video feedbacks. So happy customers. And just like to point out that we were the first one to introduce online training in drug discovery and designing, and we are also the first one to conduct online and on-site training in gene and genome editing using CRISPR-Cas9. Courses, I have already told you that we've conducted more than 70 free courses since February 2020, and then more than 14,000 people have participated from all over the world. So these are the services like virtual screening, molecular docking, MD simulations, predictive toxicology, and NGS data analysis and genome editing. Personalized training. Now, personalized training is something very important. Why? Because people who want to learn certain topic or one topic in particular, we design the program and we conduct the training on that particular program. Similarly, research project consultancy, this is especially for the students who are PhD scholars and they stuck at one point of time in their PhD research and they do not know where to go. So we design the future course of their PhD project. Last is this. You can follow us on Facebook and Instagram, and then you could see that YouTube and LinkedIn. So this, if you want, you can take the screenshot of this particular window. And now I would like to introduce the programs. Now the program Envision with BDG. Envision with BDG is a program after, like, how we started this program and what is the program. After 6 months or 7 months of rigorous efforts for providing free courses and to give more to the society, we thought that why not to do something new. So that's why I contacted people on into my connections, and a lot of people, in fact I should say that mostly all of them have replied, and then we started this program. The aim was that we should conduct 2 hours or 60 minutes or 90 minutes session on the topic of their expertise. So these are renowned professionals from their field, and definitely you will be amazed to know that in just a time span of the last 10 days or 15 days, we've conducted more than 4 or 5 sessions. So this session or today's session will be conducted by Dr. Nagagireesh Bojanala, and basically he's a professor and dean of research at Ramaiah College of Science and Commerce in Bengaluru, India. And he works on developmental genetics in the small nematode, that is Caenorhabditis Elegans, and teaches molecular biology, genomics, and immunology courses in the past. He has worked as a postdoctoral fellow at IISc, that is Indian Institute of Science, Bengaluru, and TIFR-Mumbai, and Institut Curie, Paris, France, and Penn State University, that is United States of America. He has been awarded the prize for postdoctoral and travel grant from Institut Curie, Paris, and full scholarships to pursue his master's at McMaster University, Canada. So now I would like to hand over this session to Dr. Nagagireesh, and thank you very much for listening to me and giving me time. And Dr. Nagagireesh, you here?
I'm here, Asif. Can you hear me? Am I clear and loud?
Yes, perfect. You are here.
Okay. So can I go ahead and sharing the presentation?
Yes, please go ahead.
Yeah, 1 second. Is the presentation visible to all?
Yes, it's showing that you are started screen sharing.
But it's still rolling. I don't know. 1 second. Okay.
Yes,
So,
go ahead.
I'm unable to go to the full screen mode. Okay, now it got it. Maybe better. Should I stop my video? Maybe that may help us with the connections and all. Okay. Okay, good evening, everyone. Is the slides visible? I'm loud and clear. Hello?
Yes, sir.
Yes, perfect. It's perfect.
Oh, okay, Asif. Okay. Good evening, one and all.
Yes, it's very clear.
A nice introduction, Asif. Good to see the activities and help coming from for the students from the BDG. And it's kind of night time, but hope everyone is motivated to follow up on the talk. Just introduce the topic. Maybe already you have known. I will be talking about the Caenorhabditis Elegans, an elegant model to study developmental biology. 1 second. Anura, go out, please. So as you look in the title can be divided into 3 different concepts. For example, I will be covering main 3 areas. The first one, as suggested, I will be talking about an organism, a small nematode called Caenorhabditis elegans, and I will be connecting it to a specific biological phenomenon called developmental biology. And why I'm connecting it, why I'm talking about why this organism is an elegant model, that comes to the point of we should speak about what are model organisms. So thus the whole talk today will be connected into 3 terms, 3 concepts. Caenorhabditis elegans, what is it and why should we study about it? And model organisms, what are they? What are the advantages? What answers these model organisms are giving to scientists all over the world on their specific biological question? And what is developmental biology? And why should we study this specific branch of science? What is it? Why is it important? Those kind of areas I will take forth. Individually, I will take into a little bit more broader details. And at the end of the session, we will take up into more queries or any discussions from the group. So let's take one first, Caenorhabditis elegans. It is commonly known as C. elegans or the worm. And it is a simple nematode. It's very simple. It is a non-parasitic nematode species. It doesn't have any parasitic inclination toward diseases. It is a non-parasitic nematode species. And it is a very small organism. For example, it's 1 millimeter in length, whole animal from anterior to posterior. It measures 1 millimeter. That's why it's difficult to look for the naked eye. That's why we have a special microscopes, compound microscopes, so that you look at the organism at the higher magnification level. And the whole adult animal, it consists of only 959 somatic cells. It's not about, if you compare humans, billions and trillions of neurons and cells, excuse me. And this animal has only 959 somatic cells. And it has a very primitive nervous system with 302 neurons. I will talk much about the organism a little bit more. And coming on to the life cycle and development of the organism, you can see that it's an adult animal and it exists in 2 sexes. One is hermaphrodite, one is male. I will talk further in the next few slides. Coming on to the hermaphrodite, it starts self-fertilization within itself because it has eggs, it has sperm. And when the fertilization happens, single cell is it starts development inside the mother itself. It starts with a single cell division, followed by a specific developmental pattern called in utero development, approximately 40 minutes. And then it later, excuse me, slide share is going for auto. So later the eggs are laid from the uterus. They will go into a specific developmental fate till hatching, and it becomes larva L1, L2, L3, L4, and it becomes into adult. Each life cycle, each stage has a specific developmental timing, specific growth timing. And as I said, it has 2 sexes. One is hermaphrodite, one is male. And the hermaphrodite is denoted by the notation XX in terms of the chromosomes. And the male is called as XO because there is no Y chromosomes in the worms. The male exists with only 1 X chromosome. And usually when you look morphologically, this is the hermaphrodite. The body looks in terms of the pharyngeal for helping in the digestion and ingestion, and the excretory system at the end. followed by only few major systems. For example, this is the one in the middle you are looking is called gonad. It's a bilobed gonad. Majority of the body is covered by reproductive system. These are the developing oocytes, followed by spermatheca, and the fertilized eggs are here. And there is a specific coelom and also specific intestine that helps in digestion here. Basically, the system has very simple developmental systems in the animal. Coming on to the male, there is sexual dimorphism as seen. The hermaphrodite has a tapered tail, and the male has a specific male reproductive organs as suggested here. There is a male copulatory spicules, fan, rays, and specific spermatheca development organs are here. So when you look closer, the organism in terms of scanning electron microscope, you look more closer as the animal is 1 millimeter. You look in the scans, you get into a specific morphology. And as I suggested, the male has a specific tail and the tail has a bulbous organism. So 1 second. Somehow this presentation is going on auto mode. Can you give me 1 second? So slideshow. Timings. Okay, are we in? Hello?
Yes, sure. Please go ahead, sir.
Yeah. So that's what the animal looks like that. It's a simple nematode. Then we come back to why is it? Why do you study a model organism? Why it is called as a model organism? What are they? So you come back to again in terms of what are the common features of model organisms. For example, if you probably the whole world knows about the vaccine development process. Even my daughter talks about corona and vaccine. And and it's clear that to develop a vaccine, it takes human clinical trials. So if you want to study a disease, find a cure for a disease, you need to use humans as a specimens or subjects for understanding the disease and finding a drug target for that. But is it available? Is it cost effective, or is it ethical to use humans always for whatever query, whatever concept you want to study or whatever disease you want to study? But it is not. It is not actually ethically. There is considerations into that. There will be ethical consideration. That's why we want to find something surrogates for humans where we can use the surrogates and then look into developing a specific target or looking into a more basic fundamental aspects of a specific diseases. That's what the advantages of model organism comes, because when you look into the evolutionary scale, we have grown from small organisms. We evolved from simple organisms into human beings right now here. So we have a commonality that we share along that we will talk in further. So the most simple common features of model organism, to use it as a surrogate for human being, the first thing is that it should be simple. It should be simple to understand, simple to study. At the same time, as humans, we have a lifespan of 80 to 100 years. But if you want to study a kind of genetics, you want to look in a generations, humans in a kind of pedigree, we look for grandmother to granddaughter, that's it. A researcher can spend whole 60 years looking into 4 people from a family. It's not possible. That's why when you look for model organisms, you want to have more genetics, more data, more analysis, more statistics. Then you go into a specific model organism that has a short life cycle as possible, a rapid development. That means it can go quickly, it can give babies quickly, and they can develop quickly. That is very important for model organism. At the same time, ease of access. How can you access it? How can you grow in the lab? How can you manipulate it in the lab? That's also really important. For example, you need a little bit of small workspace because establishing a lab is very costly. It's like you need to have grants and budget. But if you can grow them in a small workspace, smaller space, you can get a lot of work out of it with a limited budget. Then that is the best feature you're looking for a model organism. Okay, the presentation is somehow holded. If there's some admin, somehow I am unable to move the slides. Asif?
Yes, Dr. Nagagireesh, tell me.
Somehow the slides are not moving. Maybe, okay, I will start the slides are got stuck. Okay. Okay, I'm resharing. We'll see how it works. Okay.
It's working, right?
Yeah, it's working now. It's okay.
Okay, yeah, please go ahead.
So that's what you're looking for, a model organism. So have you known any model organism? Have you studied about them in your biology studies? I can remember when I was drawing some diagrams of Hydra. I can easily remember one phenomena called phenomena of regeneration. The Hydra has the potential. You can chop them with the knife, two parts, and one part develop into a completely adult and functional Hydra, and the other part grows into the tail. That was a very fascinating thing. I was very motivated, curious about to see what happens during my high school stage, how the Hydra can regenerate. That's a phenomenon of regeneration in Hydra, but there exists more model organisms across animal phyla. People and scientists are studying butterflies, mice, fish, worms, whatever features they feel comfortable, and they find answers into a curiosity, they are studying on them. And majority over a decade, there are a lot of model organisms, but many tools and resources have been developed for some of the best ones. For example, you have an idea about mice research, fruit fly, Drosophila research, xenopus frog, fish, zebrafish. At the same time, you can look into C. elegans, and yeast is also a good model for biochemistry. And why do we prefer one organism, the other organism? It depends on what the question the researcher is looking for. For example, when a researcher is looking for to study an embryogenesis, how embryogenesis happens, he doesn't need to look anywhere, need to get big and costly microscopes. He can simply grow some fish in the aquarium in the lab and then take and look in the eggs, because they are so translucent and transparent. You can literally follow the cell division cycle, how the larva is forming up here. At the same way, each model has its own advantages. For example, if you want to define specifically, as discussed in the first 3 points, the model organisms are vital tools used by researchers around the globe. These organisms share many genes with the human. One of the important concepts why I want to study a small nematode or a fly that feeds on fruits, because it belongs somehow, the genes it shades, my genome is shared with it. I evolved from it only. If I want to study a specific gene, I can go back and look at the genes rather than humans study them in this model organism. That's why they have been given so much importance in research in biology and a few different disciplines. And as I suggested, they are easily maintained in the lab, short generation times, and they are amenable for genetic manipulation. That are the concluding things about model organisms. Then specifically coming on to the point of the nematode, Caenorhabditis elegans, why I want to use C. elegans, as suggested with the zebrafish transparent embryology, C. elegans compared here in the table with the yeast, Drosophila, and rodent mice here. I will highlight in terms of the C. elegans what features it holds, as suggested briefly in the first few slides. It has a short lifespan. Its life cycle is rapid. In 2 weeks, it will grow and it will die literally in 2 weeks of time. But by that time, it gives a lot of babies, small body size, transparent body, ease of genetic manipulation. And you can do gene knockout technology, as you already known by CRISPR, gene editing technology. Same way, C. elegans, a phenomenon called RNA interference was discovered. And this phenomenon revolutionized gene silencing in many of the biological questions. And also, given the neurobiology and given the simplistic neurons, it is a very good model to study behavioral pattern. So same way, you can see some of the advantages for each the model organism. As suggested, most genes in C. elegans genome have functional counterparts in the humans, either orthologs, homologs, and in similar way, some of the function, how a cell undergoes into cell cycle or cell division, cycling genes control this phenomena. They control cell cycle control regulation in the C. elegans in the nematode. They control similar way in the humans also. And also it is a good model to study basic understanding of several disease models. For human diseases, I will talk about a little bit briefly at the end of the session. and it has a lot of mutants that can give a better understanding of neurological disorders, heart diseases, and kidney disease. That's what it. But when you want to see in phylogenetically, mice is a bit closer to humans compared to the worms, but they all share a percentage of the genome with humans. As for example, the C. elegans codes for almost 18,000 genes that can code for approximately 25,000 proteins. So some of the proteins are similar to functionally humans that we will speak in few at the end of the session. So we covered so far what is C. elegans, and we covered about common features of model organisms, why a C. elegans can be a good model. I spoke briefly, but I will extrapolate this thing a little bit more details in terms of cell biology, genetics, molecular biology, RNA interference, and diseases. I will talk a little bit more. But as suggested by my title, it is a good model for developmental biology. So why I specifically chose that topic, why I want to speak more on this, why should we study this? My perspective was, as I suggested, I was fascinated by the Hydra diagrams looking in my biology books, and I was very curious to understand developmental biology because I started as an adult from a single fertilized egg, that is single cell, that grows through 9 months and gives a baby. From the baby, I become an adult, and certain stage, my differentiation, my growth all stop. So it's kind of self question I was looking into how this happens, how cells communicate with each other, how they grow, how hands form, how eye forms, those kind of fascinating question I was looking into. So that's why I decided to study developmental biology, and almost a decade of research I pursued in that all over the world. And I can say that my passion and curiosity always starts from fresh. I can take one question from undergraduate, look into some concept. I feel like I can start over again. So I can briefly give in few slides why I like developmental biology. Simply, we go in next few 3 slides. Here, I'm showing into 3 different organisms or 3 different phylogeny based organisms. For example, plants, animals. In the animals, I'm talking about insects and human beings here. So if you look in the developmental process of this, there is a kind of a single cell or pupae, group of pupae here. They are going through a process of development and they are going to a next stage. There is a process, there is a stage. One stage grows and proceeds into second stage, and it goes into a stage here. This stage is called final stage. This is terminally differentiated state. Growth has been done. It's a differentiation pattern has been formed for a single cell into different cells. And this pattern is again diverse. For example, here you're starting with single cell and you're going into the adult here. So basically, you look in more details here. It seems like I'm admitting someone, it's getting hanging. Okay. So the presentation is hanging once I start admitting someone.
Dr. Gireesh, you just focus on the presentation.
Okay.
The technical team is there. They will admit anyone or they don't want to admit. So just please focus on the presentation.
Okay, I don't want to miss some of the students. It's okay. Yeah, I will focus from now on. I might have to reshare again.
Because the thing is now that if you will do this, then your presentation might get hanged. So that
yeah, true. Okay. Okay, it's going on. So basically that's the process. We looked in that. For example, if you talk more into a human development, for example, how I have been developed. So there is a kind of specific pattern goes with the different stages of processes. And at the end, I get into an infant, completely developed adult. But if I look more into kind of fundamentally at the cellular level, I'm starting as suggested a single cell here. And there is a specific process of cell division, right? It is a very regulated process that controls the developmental biology. A single fertilized cell that undergoes a cell division, first cell division, and it gives rise to 2 cells. Later, the 2 cells will go into one more round of cell division. They multiply, they give into 4 cells. So basically how one becomes into multiple cells through a pattern of controlled cell divisions, controlled cell cycle. That is an important. So to conclude in few terms, One of the major concepts in developmental biology is that there is a process at every stage. There is a process. The cells, they know what process they have to go through. And at the end of the process, they will develop into a specific pattern. At this stage of the process, they have a different pattern. Then they move into a next stage of process, they will have a different pattern. And there is a terminal pattern. So they know their pattern, they will go through the process, they will finish. And you look all along the animals in the phylogeny, evolution, I'm talking about plants, insects, and humans, there is a commonality. That is the beauty of developmental biology, that there is a commonality here, stages starting from cell cycle, there is a commonality in the pattern. That is the beauty of the developmental biology, process, pattern, and commonality. And not only cell cycle and cell division, one other aspect that really fascinates me through developmental biology is that morphological diversity. For example, I am human, I'm diverse from a cat, I'm diverse in terms of morphology from a dog, or you look more into a little bit deep into sea animals. There is a lot of sea animals you see, they are sea dwelling animals. You see a lot of morphological diversity among them. And you go a little bit more closer, the morphologically diversity gives a beauty also. For example, if you look, you may feel that this is a developmental results of a developmental biology that went through a morphogenesis phenomenon you consider as a beauty. And at the same time, same phenomenon, same biology, I consider beauty in other terms. This looks to me more beautiful than that. So it's an individual perception, but this beauty, that beauty, both have gone through the space process of developmental biology, pattern, and morphological diversity. So how this happens, that is one of the biggest questions. We know some answers, but we don't know yet. Many of the questions arise, and scientists all over the world using different models when they want to focus on developmental biology, they're studying in it. How morphogenesis happens, for example, during development. We start during the process of development. Here you're looking up the arrow growing up in terms of the growth. We start with an individual cell or group of cells, and they will become into a different cell types or tissues, and the tissues go and gives us to different architecture or morphogenesis of different organs. At the same time, at the end, they generate the morphological diversity. But these are different steps. How these steps are executed, for example, each cell or group of cells at certain time of development, they acquire specific kind of fate. The fate, maybe you become hand, you become an eye during the course of development. So once they acquire the fates are executed by specific phenomenon in cell biology called cell differentiation, and they gives rise to a specific kind of organogenesis or morphogenesis at the final. Then how is this execution is happening? Once a fate acquired, how it executes, how differentiates into a final form. The execution of the fates happen by specific pattern of cell divisions, pattern of cell migrations, pattern of cell fusions, or one concept you describes in fundamentally as collective cell migration. This is the events that happen at the cellular level. So that's how the morphological diversity, how the development in a controlled, coordinated fashion execute this fashion is amazing always. And there are many questions, unanswered questions in this phenomenon. So to conclude, beginning a little bit on developmental biology. So developmental biology is a science that investigates how a variety of interacting processes, for example, interacting cell or processes, generate an organism's heterogeneous shapes, diversity, size, and structural features that arise from a trajectory from embryo to the adult in a coordinated manner, or generally through a life cycle. This is in simplistic terms, you define developmental biology, but it looks as simple in the abbreviation here, but it's a very complex phenomenon, coordinated phenomenon, and we need to much to learn about it. I will talk about taking C. elegans now, a little bit more into deeper as the title discussed. I talk about C. elegans, but if you look developmentally, I have explained about cell cycle, cell divisions. You can ask more related to humans, one simple question. As I said, it's a coordinated development, coordinated process. You ask a simple question, what happens if development goes wrong? The basic steps of execution goes wrong, cell cycle goes wrong, cell division goes wrong, architecture process of the hand goes wrong. What happens? What happens in them? We can take one simple example. Developmental biology is a normal controlled phenomena happening. when it goes bad, you get nothing but cancer. So cancer is nothing but abnormal development. There are like different types of cancer depending on the organisms, but it's nothing but a controlled development has been malfunctioning by some certain diseases or mutations. It becomes into abnormal development. For example, controlled cell cycle, it goes into uncontrolled cell cycle passion, then the tumor formation happens. That's what I just want to briefly talk one slide on this. When you talk a little bit more about when cancer cells spread, they need to migrate. Again, they have to use cell migration processes, and they can invade. Again, cell invasion comes there during development. That's a metastasis happens. But just to say simply, a normal developmental phenomenon by some aspect of mutation or disease exposed to environmental toxin, it can become abnormal and it becomes cancerous. You see, you're looking here, cells in the normal stage, normal cell biological and biochemistry within the cell and genomic organization, normal development and architecture. When in abnormal conditions such as cancer, cytoplasm shrinks and the single nucleus becomes multiple nuclei, nucleolus becomes a much larger, multiple, and chromatin becomes a little bit coarse. That's what the development is really important to have a normal human being and normal life cycle, normal disease, normal development. Otherwise, you go into abnormal disease. So that's what we've spoken so far about C. elegans, what is a nematode. We've spoken about model organism. We've spoken about developmental biology and why should we study. And now I will take a little bit more 10-15 minutes on what is C. elegans, why it is a good model in terms of studying cell biology, genetics, biochemistry, molecular biology, RNA interference, gene knockout technology, or making transgenic animal. I will cover each concept in 1 or 2 slides and look into more details. So as I suggested, it's a very small animal, and usually in normal environmental condition, you go to your garden right now or any soil environment, you can dig some soil, you can take and put them on a Petri plate. So you can easily find the worms. These are ubiquitous. They are growing everywhere. The nematodes are available everywhere. They are small nematodes, and usually in the normal environmental conditions, they feed on organic debris. They feed on a little bit bacteria also, and they survive in the soil. But as I suggested, the model organism, when you want to study in a lab environment. You cannot study in them in natural population. You can grow them in the lab. Over 3 to 4 decades of research, people have studied the animal's physiology, growth, and maintenance. They have established a specific protocols, specific temperature for growth maintenance. That's what, coming to the point, we grow the worms in the lab in a specific nematode growth medium agar plates. So you see, there is a plate already. The agar here is being poured out and it's been dried, and later, and we grow them. We feed them specific strain of bacteria called Escherichia coli. That is called OP50 bacteria. Once the nematode growth medium dries, we will put the bacterial some small drop of the bacteria here. Given the binary cell division, within 24 hours, the bacteria grows, multiplies. A thick lawn of bacteria will form here. Then, depending on how I want to grow and maintain the worms, I will start sometimes when I have a single clonal population I want to maintain. I take a wild type strain that is called N2 or Bristol. It has been isolated from Bristol, UK, and is called as a strain called N2. This is the wild type strain, reference strain. We use all over the world, whoever have C. elegans research and C. elegans labs running in the research field. So we pick one hermaphrodite, put them here on the day 0 here or day 1, and then slowly it grows, it feeds on the bacteria, it grows. And as I suggested in the reproduction, it lays almost 300 eggs within the life cycle. And all the 300 eggs will come up in the eggs. It will hatch the eggs. And within the lawn, the egg starts to grow and become larvae 1, 2, 3, 4, and they become into adults. And within a 1 week, 1 adult becomes into 300 eggs. 300 eggs within 3 days of life cycle, 300 mothers. So basically 1 mother, 300 mother becomes here. And again, within 3 days, they will lay 300 eggs. You take the mathematics now. We are looking into a plate starting from day 1 here, 1 animal, 1 hermaphrodite. In 12 days or 2 weeks time, you are looking with a plate of 10,000 worms per petri dish. And you are saying, as I suggested, I'm putting only N2 here, or I may grow one mutant animal here. So by doing so, what I'm saying, I'm maintaining at the genetic pool, homogeneous genetic pool of worms in one single plate. So that is the beauty of genetics that gives a lot of grip to you when you're doing genetic studies. That's what you are seeing. And the bacteria may stay almost 2 weeks if the animals don't feed it. Once the animals grow a lot of babies, they will eat out the bacteria and you might have to transfer them into a separate plate. That's what the process of growth and maintenance we do in the lab. And normal growing condition for the worms is 20 degrees. So we have to maintain them in a special BOD incubators at 20 degrees. We take them into a plate, each plate or 10 plates, stack it up, one strain. We rubber band, we wrap them up, put it in a box. Like that, we can maintain tens and hundreds of boxes in the incubators. And this is the normal cycle at 20 degrees. It takes 3 days. But if you temperature, you can cool it down. You make a BOD 15 degrees incubator. The worm that becoming from egg to the adult in 3 days, it may become egg to the adult in 3.5 to 4 days, so that you can extend little bit life cycle or slow the life cycle here in 15 degrees. But at 25 degrees, you increase the temperature. Rather than 2.5 to 3 days, it grows into the life cycle from egg to the adult in 2 days itself. That's what you can control the growth also based on the temperature. And in terms of the maintenance, you can freeze them. For example, you can just freeze a specific freezing protocol solutions. You can freeze them at -80 specific bacterial vials. You can put it into the -80 freezers, or you use a special protocol and you can freeze them in the liquid nitrogen also. So -196 degrees centigrade also the worms will resistant for the cold conditions, and they will stay dormant there. For example, I put all these worms in a vial with a specific chemicals and I freeze them along with the bacteria. I put it in a liquid nitrogen freezer. Okay, I'm putting it October 16th, 2020. And 20 years later, I come back. I'm going into 2040 October. Hope COVID will not be there at that time. Then I'm taking a specific vial I froze. I'm taking the vial and putting into a fresh bacterial agar plate. You just give 1 minute or 30 seconds. You can come and look in the microscope. You can find a moving worms. This proportion, a percentage of the worms start moving into that. And they come back into the bacteria and they start getting back into the normal life cycle, eating and growth, and they start making babies. That's what the beauty of you can cryopreservation, and still how the worm, such a small nematode, can survive over 20 to 30 years and regain its life back. That is an amazing thing people are studying now. So as I suggested, that's what the growth and maintenance of the worm. And I just want to briefly talk about, as you may be choosing your field of research in the further higher studies as PhD. So I just give you brief 3 areas where the C. elegans can be used to study any of these PhD plans or biological questions. So I will talk a little bit more about cellular features, what the worm at the cell biology level, what are the advantages of the C. elegans to study cell biology, and then I will go into genetics, or I will go into genomic resources a little bit, RNA interference, and then I will go into specific molecular biology. In terms of genetics, we can talk about forward and reverse genetics also. Okay, so coming on to the point of cell biology. So as I suggested, it's a very transparent animal, just like zebrafish. When you have a simple light microscopes, you can take the egg on a slide, you look at the egg morphology. You can take a single animal, hermaphrodite, you make a slide. You can look at the morphology of the animal in a simple light microscope. And one of the beauty of cell biology for the worm is that there's 3 group of researchers or 3 individuals. They were really fascinated about the small number of cells the adult worm has, around 1,000 cells. And they have seen the transparency of the egg. They started with a single zygote, and they followed that one egg with 1 cell become into 2 cells. Then they started tracing the lineage, tracing the cell lineage pattern. Okay, 1 become 2, 2 become 4, 4 become 8, 8 become 16. Then from this 16, which cell is become into hypodermis, or they may become into a ball of cells here, embryonic cell division, at a certain time. For example, starting with the 0 development cycle, 10 hours, 20 hours, 30 hours, 40 hours, 50 hours. That means around 2.5 to 3 days. It starts with embryo L1, L2, L3, L4. But at the embryonic and cleavage divisions at certain time, the cell cycle division pattern stops. After it stops, there is a kind of little bit growth and pattern formation will happen. For example, I want to study in the adult a muscle, and I want to see that the muscles or locomotion body muscles may be here. I want to see that where these muscle cells came from the uh, zygote here. Which of these division resulted in that? Same way, the reproductive system here, the vulva development, female reproductive system, from which cells of the embryo it came? Where are the cells in the L1 stage? Where are the cells located in L3 stage? I can do complete tracing. This complete lineage has been identified, just like a Bible. They have put all this directory out. We can study in take whichever larval stages you want. You can look into the cell. Okay, this cell will become into L4 and now neurons are reproductive system. I know I can trace it back. How does you can trace it back? Again, hanging. So how can I trace it back? Basically, I can trace it back simply. For example, there is a scientist called John Sulstan. He was given a Nobel Prize also. I will talk a little bit more in one slide. What he started synchronized animals. Several of the C. elegans started with a fertilized egg. So he timed in a way that he can take one egg here, whatever patients he has, he can keep drawing the cell division in a rough sketch diagram on his paper. He can draw from here and follow that this cell anterior posterior, what it has become here. It has become into mesoderm. It has become into epidermal cell. So he defined them. He followed the diagrams. He traced it back. Like that, he traced it back till to the adult animal. So a synchronized animals, batch of animals. He can stop at certain time. I can come back with another animal starting this time. So he put a lot of hard work to do all this lineage pattern. For example, starting from P0, there will be AB and there will be P1, ABA, AB posterior, anterior, specific cells up to that. Total number of cells you can count, hatching, and you can move into that. So for example, so you can see here, this is cells without the eggshell. When there is no eggshell, you can see different pattern of the cell cycle in terms of gastrulation, in terms of larva molting out, larva coming up. You can see into that in a transparently. This is again a kind of scanning electron microscope. But as I suggested, I just want to give you in terms of one phenomena called reproductive system development in the nematode. That's in the adult animal, it looks here, the central point is called vulva, and it helps in the eggs to come out after fertilization. So when you look in the L4 stage, the morphology of the structure looks like that. But how does this phenomena happen from the larval stage 1? Which cells have been given this kind of structure? How many cells are here? All you can study by simply looking into a light microscope, analyzing different larval stages. For example, I can clearly define you in the cartoon that at the L4 stage, this reproductive organ has 22 cells, 22 epidermal cells. How they formed? They formed from three different cells called P5P, P6P, P7P. They started from L3 stage. This is the L4 stage, at the end of L4 stage. So how does these cells look in L3 stage? They look like that. You can see the beauty of the nucleus, whole cell just with the light microscope. And there are anterior, posterior cells in the central, P6P, P5, P7P. These are called hypodermal cell or vulval precursor cell. These three cells give rise to 22 cells later. How they will give? They will receive a specific cell-to-cell communication signal from this gonad here. This is a gonad developing already. A specific cell in the gonad called anchor cell that releases specific ligand molecule. That ligand molecule goes and spread across as a morphogen or gradient pattern, and it talks to these three cells in a different pattern. For example, this P6P epidermal cells very close to the anchor cell, sending cell. This is cell receiving cell. It receives maximum of the signal, maximum of the ligand through its receptor. So it takes a specific fate called primary fate. That primary fate results one cell to become 2, 2 to become 4, 4 to become 8 during cell division and during larval stage. So P6P gives rise to 3 cell divisions, 8 cells. And P5, P7 are anterior mirror images. They will receive a moderate signal that is lesser than the P6P given the distance here. And they will adopt a fate called secondary fate. And they will go into 3 rounds of cell division. 1 becomes 2, 2 becomes 4, 4 becomes 7 here. You can see here 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7 cell at the end. But P6P has 2, 2, 2 to 8 cells. And they will go into a specific pattern of cell division called transverse and horizontal divisions. We will define as a lineage patterns, but it's okay. Later at certain time, all these 22 cells, they will migrate, they will talk to each other, then they will become like this in L4 stage. You look more closer in the cartoon diagram, we define the primary, secondary, and tertiary. In certain stage, they migrate up and they become into different phenomenon dividing. This is longitudinal division. They go into a transverse division and they have a different pattern to give rise to a specific reproductive organ. So this was in terms of cell biology. That is an interesting phenomenon was observed by Paul Sternberg from California Institute of Technology. Then I'm saying that this is the signal sending cell. So if you are a scientist, you have to prove that. How can you say that this is a signal sending cell? So the C. elegans gives another phenomena called, there is a laser ablation technique. I can kill this cell at the L3 stage. For example, I'm studying this development. I know that these 3 cells gives rise to the reproductive development. Whether this is the anchor cell giving it or not, to do that, I take a worm and cover the worm in the cover slip in the slide, and then I put under a laser microscope and I start hitting these cells specifically with a laser. So I can start hitting the cell with the laser. So at certain time of development, at certain time of laser intensity, I can see that this cell is dying. I can see clearly it disappears. Then I can pick this L3 animal, put it into the plate, and then I want to see that whether these cells have received the signal or they have given the reproductive system or not. So when I look in the adult, I can see that in the L4 stage also there will be only 3 cells. They have not developed any, they have not acquired any cell fates. They have not developed and divided. They have not given the 22 cells because they did not receive the grown-up signal from the anchor cell. That's what they proved. That is one more amazing technology that C. elegans can be used for. So coming on to a little bit in terms of the pathways, what is the signal sending in terms of the proteins? What is in terms of morphology of it, in terms of structures of it? And then in terms of signal receiving cell, what are the pathways are involved? It's been identified that the signal coming from the anchor cell is called as epidermal growth factor hormone, epidermal growth factor protein, that in C. elegans is called LIN3. In humans, it is called EGF. Same protein that is working in humans in cell-to-cell communication. It is working in similar way in C. elegans, that is called LIN3. And the receptor on the signal receiving cell called epidermal growth factor receptor, EGFR. In C. elegans, it is called lead-23. Lead-23 is the receptor. So the signal ligand that goes and comes as a morphogen here, the receptor receives it, and in the cytoplasm, specific signaling cascade goes, and in the nucleus, a gene expression pattern starts. This is the primary, this is secondary cells. They will have different signaling pathways. The signal has been multiplied in cytoplasm by specific cascades that includes RAS proteins. RAS protein is one of the when it is screwed up, you get a cancer. So whatever happening in C. elegans in controlled cell division, same EGFR RAS, LIN3, lead-23, RAS is called lead-16 C. elegans. They control same cell division, cell growth pattern. When it becomes abnormal, when RAS is hyperactivated, C. elegans cell division also multiplies. That also gives rise to similar to the cancer that we will see in a few slides away from that when we talk about human diseases. Okay, so far so good. You're able to understand? Hello? Is everyone there?
Yes, sir.
Yes, sir.
Okay, that's
Yes, sir.
yeah. In terms of cell development, now we talk a little bit more about genetics and genomics and molecular biology of C. elegans. So, as spoken by Asif about the genomics and resources, CRISPR, gene knockout, gene editing, and artificial intelligence. So, when people were starting the sequencing around early 1990s and so this is the first vertebrate, the first animal that has been picked up for whole genomic sequencing. So, in 1998, they started. They have taken. They wanted to sequence this small nematode. They want to know about the whole genome of it. They did completed the project in 2002, and they identified that the genome of the worm is around approximate 100 million base pairs, and it codes for probably this is the data is old. Now the genes are around 18,000 genes. They code mostly about 25,000 proteins. When you look and compare the genome to humans, almost 40% homology you can find for the humans in this animal. Okay, Ruman, you have any question? You raised your hand. Umay Ruman. Okay, so going into the genome, what are the chromosomes?
Pause, Dr. Nagagireesh. People, if you have the questions, then you can note down and then you can copy and paste it in the chat section so that we will address all the questions one by one after Dr. Nagagireesh finishes his presentation. Thank you.
Okay, thank you. Thank you. So that's what. So as I suggested, this is the whole genome that been sequenced. The whole genome has been divided into 5 autosomes here, chromosome 1, 2, 3, 4, 5, and there is one sex chromosome here. In the hermaphrodites, you will get 2 copies of the XX. In males, you get only 1 X. There is no Y chromosomes overall. So hopefully I want a little bit more about talk on the genetics, because the worm is a very good model for conducting genetic screens. Hopefully some of the students here have little bit background on the genetics. I will talk about 2 concepts in next few slides. One phenomena called forward genetics and reverse genetics that we will talk coming from the genome to the back. In forward genetics, what you are taking up is that you take a normal N2 strain, that is a wild type strain, you treat them with a specific chemical called ethyl methane sulfonate, that is called EMS. You are not treating, you are going into a phenomena of mutagenizing the worm. You are not going into a single worm level. When you try to do mutagenesis screens in the worms, you have to take a large population of the worms, specific concentration of the mutagen. You put them in one Pendoff tube, collect the worms, add the mutagen, and follow a specific protocol and specific timeline. After certain time, the mutagen does its job. It breaks the DNA and it induces mutations in that. The mutations may be 2 kind of phenomenon. One is point mutation, where a single nucleotide change, for example, GC in the N2 may become into AT, that is a mutant form, or it may become into a deletion, translocation, duplication, rearrangements can also happen. Not the single nucleotide, a large chunk of the DNA can also be taken out. EMS is the one that gives usually point mutations, but other way, X-ray, gamma radiation, other radiation in the worms, you can create large deletions also. So how the screens are done in the worms? Basically, as I suggested, I'm taking a specific worm, mutagenizing them. When I do this mutagenesis, when I'm incubating the worms with a mutagen, we are taking the adults that will have already developing oocytes, sperms, and development of oogenesis and spermatogenesis happens at the time. We take the young adults only, not the gravid adults, that means where they finish the embryogenesis, because when you want to mutate, you want to induce them in the oocytes or sperms to induce the mutations only. So we take specific cohort of the animals. This is the wild type plus and plus. We treat them mutagens. Then we recover the worms. When it already induced the mutation, we may initially get heterozygous animals in the F1 population. You may have a mutant copy and wild type copy. After that, you go with the Mendelian segregation pattern. The mutant over plus over mutant chromosomes, they segregate independently. And when you grow this kind of animals, there is a specific population that you may get a wild type animals in the 25, the plus plus goes together when you are crossing because it's hermaphrodite, mutated sperm, mutated egg, or an individual sperm with a normal egg. They cross each other, then plus plus 25% you get genetics. You get heterozygous animals M over plus, mutant over wild type, that is 50%. And 1/4 of the population coming from this mutated animal will be completely mutant. When you get both copies of that, then only the mutant phenotype will come up. That's the beauty of the C. elegans. You carry on the screen, you can get the mutant not in this generation, not in this generation. You will get from parent to F1 only in the F2 generation. You get into both 2 copies of the mutation and you get the mutant phenotype. For example, usually the wild type goes into a kind of snake-like fashion. It keeps walking in the bacteria like snake. But when a mutation happens in one of the where the genes control locomotion, that's called uncoordinated or unc-mutant, the worm, it will stop moving. It will become like this question mark shape, and it will stay there in the bacteria long, because once the mutagenesis happened, the gene responsible for the locomotion has been mutated, then it becomes like Unc phenotype. We call them as Unc phenotype because the worm is not moving. It's giving same question mark like kind of phenotype that you look in the microscope. So basically that gene is mutated here. That phenotype is called Unc. So taking a wild type animal that goes like snake, you physically treat them in mutagen, and look at the end of a mutagenesis. In the same plate, you will have different mutation carrying this, different worms, wild type worms, mutant worm. This looks like Unc. I will show a few more phenotypes in the coming slides. This is the screen happens. I think we can skip this slide. It's a long, tedious process. You need to have a lot of plates grown in the worms, almost 10,000, 2,000 plates to 10,000 to 20,000 plates you will be screening physically. For example, maybe taking the wild type nematode N2 here, I want to identify genes responsible for vulva development. How I can go with that without knowing anything? I can take the forward genetics approach, take the wild type worm, hit with the mutagen. I just want to see that where there is a reproductive development defective mutants. For example, I am showing here, I treated them with EMS, and at the end of that, when I isolated the mutants, I see that whatever the mother laid 300 eggs here, they stayed back in a single mother, and something happened, something malfunction, something mutation happened, reproductive system, the opening did not form normally, the eggs are not laid. So what happens in that, all these 300 eggs will hatch into larvae, and the larvae will be there growing up inside. Basically, they need the food to grow and come out. They don't have the energy, they don't have the food. What they will do, basically, they will start eating the mother. And at certain time, they will break the cuticle of the mother and they will rupture. They will come out of it. So this mutation and this mutant phenotype, we call them as vulvaless or egg-laying defective animals. So this is a forward genetics way, physically looking at a phenotype, guessing on which reproductive organ or which pharynx has been mutated, what the function is coming, either reproduction or coordination, neurons or locomotion. You define a specific phenotype, you define a specific mutant. For example, I will show a little bit more on a few mutants here. In terms of, I'm talking about body length here. I'm talking a wild type animal here. When some mutation happens in the growth and body length on that, I can see 3 mutants on the plate coming up when I did the EMS. 3 mutant animals, they look completely different from the worm. For example, the normal wild type animal. You compare this animal to this animal physically on the plate, this is double the size of the animal. Something happened in the body length maintenance during development, some mutation happened in the screening, in the mutagenesis. I can see a specific longer animal. I call this phenotype as long, LON, long. And there is another version opposite of this. There is a stout, short, and stumpy animals. They look like very small. I call this phenotype as dumpy. This is long, this is dumpy. And there is intermediate. We call them as a small animals. These all 3 phenotypes are 3 mutants coming from single wild type animal after mutagenesis. So this is the way forward genetics happens. Then you take this animal, you find out in the chromosome, you find out the gene location where the gene is mutated and identify a specific gene to that process. And at the same time, you are looking at one more phenotype called roller phenotype, where the wild type worms go in S-shaped fashion all over the lawn. This roller animal has a mutation in some neuronal and muscular coordination. It will try to go like a snake, but it's a mutated version, right? Some of the genes are not working. So it keeps rolling on itself in one place only. It may just like keep rolling on here, slowly a little bit move away, rolling on here in the lawn, but it can never carry the wild type movement, snake movement here. This is called roller. If you go to YouTube, type C. elegans roller, you can see very nice, beautiful video of how it looks on the plate that can happen, and look into that. Same way, different mutations, you can look into that. So this is a concept of forward genetics, what we are talking about. But if you have an idea about reverse genetics, it is opposite of forward genetics. For example, let's say I located the mutant or gene here for dumpy on chromosome 2. I know that dumpy 30 is the gene that controls this wild type phenomenon. When it is mutated, this dumpy will come. I know a gene called long or LON 25 that is in chromosome 4. And if the gene is normal, it looks wild type. If the gene is abnormal, the animal becomes long. That is the way forward genetics, right? But what we already know that I told in a few slides before, the whole genome of C. elegans has been identified. Whole chromosomes are mapped, genes are mapped. There are tools now. So I don't need to do tedious kind of screen to identify something. I can simply go and take the genome through bioinformatic tools. I can go back to chromosome 4, chromosome 2, chromosome X, a specific gene I'm interested. I can knock down the gene using a specific phenomena of RNA interference. From that, I can go back and look the worm. How does the wild type worm treated with a mutated treated with RNA interference or bacteria that induces gene silencing, how it looks in the mutation. So that's what called reverse genetics, gene silencing by RNA interference in C. elegans. That's been well established. How I can silence a gene. So for example, as I said, almost 20,000 genes are there in C. elegans. Researchers have identified this phenomena of RNA interference. I will talk in the next slide how it works. But to make it simplistic, researchers used molecular biology combination. They developed a specific bacterial strain called HT115. So this is artificially taken strain, a bacterial strain. It has a selection marker ampicillin, and all the genes of the whole genome, for example, 20,000 genes of C. elegans, for each gene, they have made one bacterial clone, HT115 for gene 1, HT115 for gene 2. Like that, they made a whole RNAi feeding library. So they have 20,000 genes library cloned. And then they have a specific, this is the insert here, carrying the gene of interest. And they have a specific promoters where they can, if you want to induce more of this gene product to come up, you can induce by T7 promoters in a normal way, or you want to double the production, double the transcription, you can use a specific chemical called IPTG, isopropyl 3-D1-thiogalactopyranidose. It is a lactose homolog. When you put this T7 polymerase, you can put into the same bacteria, you can induce the bacteria, adding some specific concentration of IPTG onto the plates where you grow the bacteria. This production, expression of this product in between goes exponentially. What is the product coming? This is the DNA. It can give rise to specific RNA also. For example, hopefully, I don't think so if I can ask a question in between. How gene silencing happens, we will go back into the next slide and come back into how it happens in C. elegans. For example, RNA interference. It's a very well-known phenomena. It works in plants, it works in whole animals, organisms. It's been first discovered in C. elegans, in plants also. What happens in this, that when you want to delete a specific product of the gene, when you go through central dogma, DNA gives RNA, RNA gives protein, right? The gene is the DNA. When gene is mutated, there will be no RNA, there will be no protein. But if you don't want to mutate the gene, but you can do in between some steps, you can kill the RNA again and you can stop producing the protein end product here. That's what the RNA interference mechanism comes. For example, specific gene of interest, you can design a specific double-stranded RNA for it. Then a process of protein steps in RNA interference goes through a specific protein called dicer. The dicer comes and takes a specific double-stranded RNA molecule. It cleaves them into two single strand, and there is RNA inducing silencing complex protein comes up. It replaces. It's splitted by the dicer here, the cleavage of the single strand. Then later, this strand can complementary go and bind to the genome wherever your mRNA is there. For example, this is the bacteria carrying the strain. The bacteria you're artificially inducing the double-stranded RNA through bacteria. And certain time, it becomes single-stranded. When you feed this bacteria to the worms, this specific portion of RNA can go and complementary bind into a specific genome of the worm. And when it binds, there is a specific breaking mechanism happens, specific degradation happens, and the product of the specific mRNA complementary to this gene will be completely degraded. So there is DNA, there is mRNA production already there in the worms. Binding happens complementarily. At complementarity happens at certain time, completely this mRNA will be degraded. So there is no product. Protein will not form. This is the way C. elegans RNA interference works, and it works in the cytoplasm of the cell. This is the nucleus. This is the cytoplasm where this mechanism happens. So basically what I am telling is that I'm already made this RNA library of specific gene into HT115 bacteria. Instead of the OP50, I told Escherichia coli bacteria will give the worm. I feed this HT115 to the worm. For example, gene silencing by RNA interference in C. elegans. I can go into three ways. I can either develop the specific strain of the bacteria that carries the gene of interest that produces the mRNA. I feed it. Once the worm feeds, then the bacteria can give the single-stranded mRNA. It goes through all along the worm. It goes through specific mRNAs where into individual cell cytoplasm, where it can find complementarity. It can knock down the specific mRNA there. It is a phenomenon through feeding. Or I can dip the worms in a solution of this bacteria carrying this specific strain of that. I can soak them, or I can directly soak them in double-stranded solution, or I can grow this through molecular biology, grow much of this DNA or mRNA or double-stranded RNA. I can inject them specifically into the worms, into the gonad. While injecting, I'm directly putting the chemical here, and specific complementarity can happen. The gene silencing can happen here. That's the way it was being developed in C. elegans. It's a well-known phenomenon. And also, if you have an idea, the beauty of the C. elegans taking the gonads, not only gene silencing, the same mechanism of introducing the foreign DNA into the worm gonad, we can go into making transgenic animals. For example, I have a wild type animal. I can make it glowing into green. I can make it glow into red in a specific portion of the body of the animal. Maybe I can make the pharynx red because I know specific gene or protein expressed in the pharynx. I know specific protein expressed in the reproductive organ. So I can artificially make the protein to glow like green, glow like red. How can I do that? Because we use reporter gene studies, reporter constructs, where you can inject a solution of the artificial. Excuse me. Anyone has a query? Okay. So that's what we can induce the artificial reporter genes into the worm. So once you inject and recover the worms into that, you can find the transgenic animals compared to wild type using the microscope, fluorescent microscope. Specific gene or protein you inserted into the reporter can give you a specific expression pattern here. For example, I am showing here a protein called adenine junction marker 1. And I fused this gene of interest to a GFP, green fluorescent protein, sequence. So I made an artificial reporter here, and I injected the reporter into the wild type worm at certain stage, and then I recovered the green glowing from larvae, and then I can easily, this once it carries the reporter and it gives them to the babies also. All the babies carry and they will glow at specific cell-to-cell junctions. They will grow like that. Same way, you can pick different neuron markers, neuron proteins, specific toroids of the vulva formation. You can use a adherent junction marker only. The larval stage L4, it looks like that, completely toroid-like shape. It gives a specific pattern. And neurons you're looking at here, you're looking into a seam cell locomotion, several things you look into that. And coming on to vulva formation, that was my PhD and postdoctoral research. I'm showing this green glowing worm where it is an anchor cell here. I was studying a specific gene called NHR25, nuclear hormone receptor. It was involved in the development of the vulva formation. You can see the expression in throughout the development of the animal, not only receiving signal sending cell also. This is the anchor cell, and at certain time the expression of my gene stops in this cell, and it comes in the signal receiving cell, and it comes in all the 22 cells. If you want to study a single cell level, single organism, single reproductive development, how cells communicate, this is the beauty of the C. elegans. You can study into that. So I will conclude in the next 2 slides, a little bit, hopefully we covered so much on cell biology, molecular biology, forward and reverse genetics. And as I have been speaking, so you're looking into a time scale of 2002 to 2008, over a kind of span of 8 to 10 years, C. elegans research was grown exponentially. And as suggested, it's been a lot of accolades have been come for C. elegans research. 3 Nobel Prizes have come on this 8 to 10 year span. For example, whatever lineage I was speaking in the beginning, so these are the 3 scientists. This is Sidney Brenner. He's a father of C. elegans biology. He is the one picked to study this nematode for vertebrate, invertebrate biology. And this is Rob Harwich. This is Sulstan. They studied the phenomena of cell lineage, cell death, apoptosis in the worm, and they have received Nobel Prize in 2002. And a little bit 4 years later, and this is the 2 people, Andrew Fire and Craig Mello. They are the one, they identified the phenomena of RNA interference in the worm, gene silencing, and they have been awarded Nobel Prize in 2006. And in 2008, I was talking about glowing worms, green fluorescent protein, reporter expression studies. They revolutionized in terms of disease mechanism, scanning, screening, all of the stuff with the just one small GFP protein. The whole science has been moved ahead into a lot of steps. And these 3 people, this is Martin Chalfie, one of the C. elegans researcher. He pioneered and he did a lot of work on the reporter studies. And one more CN, Japanese, and another scientist who isolated the GFP protein. They have been combinedly given a Nobel Prize in 2008. Okay, so given the time, I will quickly now try to conclude in one slide. For example, what I have been talking about human diseases, human relevant proteins for C. elegans. So just briefly, taking into the C. elegans, taking into the vulva development only, I will just conclude in this slide. These are the human proteins, EGF, epidermal growth factor, RAS here. This is another protein, RAF. This is MAP kinase. This is a transcription factor. All these proteins are involved in human cancer. And this is in terms of the C. elegans, homologs, orthologs here. You look in LIN3, that is EGF, RAS here called LED60, and they work in a specific pathway as I suggested, a specific cascade in the cytoplasm, specific gene expression pattern in the nucleus. And what happens when the RAS overexpression happens, you get a cancer. Same way, the people identified that if there is a mutation of gain of function situation in terms of the RAS LED16 C. elegans, we get a specific phenotype called multivulva. For example, in the normal wild type worm, only one reproductive opening will be there, one vulva will be there. But when a RAS signal, RAS protein mutation happens in C. elegans, gain of function situation, not only one cell, the other P3, P4, P8, P7, where P5, P6, 7 gives rise to the vulva here, right? There are few cells P3, P4, and P8, the neighboring cells. When RAS is hyperactivated, not only one opening, there will be multiple vulva forming in the animal, just like cancer, just like tumors. There will be multiple openings, multiple cells, a lot of overexpression of the cells. And rather than 22 cells, they give rise to more cells sometimes. The same signal, when hyperactivation, that cell division happens, there is multi-vulva phenomena comes in the C. elegans. The same phenomena is similar to the cancer tumor formation, whatever happens into the humans. So basically that comes to the conclusion of that. So I will briefly end the session summarizing what we have spoken today in the next few slides. So basically we have spoken about what is the C. elegans about, what are model organisms, what is developmental biology, and how we can take a small nematode and study it to understand human biology. For example, it's been very nicely said by German scientist Friedrich Nietzsche. He said that you have evolved from the worm to human in terms of evolution, but much within you is still worm. That's what I'm saying, that though I'm human, my genes are still coming from the worm. I carry similar to the nematode, small nematode, and I can study the developmental biology phenomena similar to the humans into the worms. I think that's better. I can conclude. More queries come, I can explain a little bit more on that. Thank you, BDG, and thank you, the audience, for your patience. And the session is now open for Q&A.
Thank you, Dr. Nagagireesh. It was a really exciting session. And I would like to thank you on behalf of BDG and all the participants who have participated in this program. So first of all, thank you. Now we've, as you know, these sessions are live on our YouTube channel also. So, you know, we've got some questions over there and then over here. So
yeah, sure.
The questions from here. So the first question is, how this RNAi helpful in cancer treatment?
Okay. So basically, as I suggested, the RNA interference, it's in the lower organisms, you use it as specific RNA molecule, you silence them. But you grow up a little bit more, there will be one more phenomena is called as siRNA. In humans, we go into a specific level of different RNAs. One is siRNAs, and then microRNAs, you go into that. Specifically, you're talking into the same phenomenon, but you are going into a different pattern of RNAs. Whatever comes in C. elegans, the double-strand RNA may not work similarly in the humans. They work through a specific phenomena of siRNA or microRNA. And both siRNAs, microRNAs in humans, they have been known to be involved in cancer, and you can use a specific knockout mechanism in that also. But to coming on to humans, it is more advanced technique, you call it as genome editing technology. In C. elegans, it's a simple RNA interference. But when you come into humans, as suggested from beginning, the CRISPR technology takes over into that. CRISPR acts more into that, and the mRNA, microRNA also help into knock out the cancer.
Yes, yes.
It's different phenomena. Coming on to the nematodes, it works differently, but in humans, you may not feed bacteria to them, right? You may use a specific siRNA in zebrafish, but in humans, you use a specific gene editing technology that goes into an advanced module of CRISPR. That is the one acting, but additional to that, there are a few more other gene editing technologies, not specifically a kind of what's happening in the nematode. CRISPR is the best one we can talk about.
Sure. Thank you. So the next question is, can you please explain more about gene NHR25?
Okay. Yeah, let me, my PowerPoint has been, okay. How? I had one more slide in another presentation. I didn't pick it. It's okay. So I will go to the slide I talked about nuclear hormone receptor. So NHR25 is a specific nuclear hormone receptors protein. In C. elegans, it is called NHR25. In humans, it is called steroidogen factor 1. So you have an idea about steroid hormone signaling. For example, as I said, the worms grow from egg to larva 1 to larva 2 and molting. Same way the butterfly goes to molting, few other animals go to the molting process. The molting of from larva 1 to larva 2 and the growth metamorphosis is controlled by specific family of steroid hormones that is called SF1 in humans, and in C. elegans this is called nuclear hormone receptors. They control a lot of developmental processes, and not only vulva development, they control in terms of molting also in the worm. And lot of processes in terms of male reproductive system, female reproductive system, seam cell formation in the epidermis. So basically it is a nuclear hormone receptor family belonging to the steroidogenetic factor 1 in humans, and they are also involved in for example prostate cancer. This is the signaling pathway that is abnormally working, and they are nothing but, I am saying steroidogenetic factors, they are nothing but this hormones, testosterone, progesterone, and estrogen. These are the hormone family. They control all these, belonging to the same family in the worms. In humans, these are the hormones. And in NHR25 in C. elegans, it is an SF1 in humans, or belonging to this testosterone family.
Sure, thank you. So the next question is like when we grow C. elegans, other microorganisms also grow with C. elegans. So how we identify the C. elegans?
Okay, so that's what the situation when you are doing the growth conditions in, for example, in natural population. That's why I said that we grow them in isolated and controlled environment conditions in the labs. Let's say you are bringing some worm outside into the plate, right? When you are bringing it from freshly from the garden soil, I poured it here along with C. elegans. I can find many bacteria, fungi, and a few other microbes. All are maybe growing on the plate. So that is when you bring it freshly from the soil. But in the lab, in the controlled condition, when I'm growing, I will have a specific phenomenon that I will add antibiotics. I will add specific to control non-interesting bacteria growing that. I will have this OP50 bacteria. That bacteria also has a specific uracil deficiency. It is a specific strain of the bacteria. When you mix some chemicals to make this nematode growth plate, you leave the plate outside. After 2-3 days, after 1 week, some other bacteria and fungi may grow. But in generally, whatever the control chemicals we have added into this, in 1 to 2 days, nothing grows on it. Only when I put this in a specific temperature condition in 37 degrees, make the plate, put the bacteria, I can guarantee you that in 1 to 4 days, only this Escherichia coli only grows in that. And if I have to control the opening the lid and closing the lid, I will go in a small room. I have controlled condition there. It's okay, some air goes in. But I can assure you that the starting point, only the OP50 bacteria is growing in my lab conditions. And when I put a single nematode, so what will be there in that? Only the worm of a specific mutant or specific wild type, whatever genomic I have constitution, and the OP50 bacteria, both will be there only. I can assure you that. There will be nothing growing into that unless 3-4 days. But if you don't know the conditions, you don't there will be contamination. There will be fungus and other bacteria can also go if your lab is not maintained clean. And main biggest enemy for this worm plate is called mites. Sometimes you have something growing on somewhere in the lab, so you get a mite infection. You don't use proper conditions. When I open the lid also, I will flame the lid and put it back. I will be opening this near the microscope where the Bunsen burner is on. Always my surroundings are fire is on a little bit, small fire. So when I touch the worm, there is a worm pick also. I forgot to show that. I touch and just decontaminate the pick in the flame, and then wait few minutes, few seconds, and then touch the worm, transfer the worm also. So we use a lot of aseptic conditions when we maintain the worm. I can assure you that for 2 to 3 days, if proper aseptic condition maintained in my plate, in this Petri plate, only my worms and bacteria are there. If I don't follow aseptic, I can grow most of the things. And when I do that, as I said, only 2 things are here. And whatever is growing on is the bacteria. I can easily identify them once I get trained into with Maya and using the microscopes.
Thank you. Thank you. And this question is coming from YouTube. So, and probably this is the last question which we will take.
It's okay. A few more questions I am okay to take.
So, could you please tell us something related to aging and neurodegenerative diseases studies in C. elegans model?
Okay, that's what the few slides I clipped in. I skipped in the last because I thought I was going overboard. Okay, so as I suggested, I want to talk about aging and neurodegeneration. This is a paper very nicely describes methods in molecular biology in 2018. What can we learn about human diseases from nematode C. elegans? It's been from Boston and it is from Colorado, Denver, JVR Aprad and Scott Alper. They worked on studying all these genes related to human disease in that. As I suggested, we are a bit warm inside and how we can do studies in that. For example, there is a gene called SFT4. SFT4 is the one gene in C. elegans. They were interested in neurodegeneration studies. What they have find out that I will tell about what is gene that looks in humans. They made a specific fusion reporter for this, and they tried to look in where the expression of this gene happened. They were able to see that the gene is expressed in the neuromuscular or in the pharynx region, expressed in some of the epidermal muscles and neurons here, and expressed in the reproductive system. You are looking in the wild type animal in a normal light microscope, a normal DIC microscope, and when you hit with the fluorescent reporter you make for the gene, you look for a specific pattern. It is expressed in neurons also. When it is expressed neurons in the wild type, that means it has some function in the neurobiology. So what is this SFT4 in C. elegans? What is it called in humans? When they did comparative studies in the genomes, they found out that they belonging to a presenilin family protein. And what is this presenilin? It is involved in Alzheimer's disease. It is involved in many of the neurodegenerative diseases. It's a specific morphology, specific transmembrane domain. How this looks in human, the protein at the protein three-dimensional level, it has a similar homology in terms of the C. elegans, similar architecture, similar functional expression in the neurons. When it's mutated, it gives some defective mutants in the neurobiology that is similar to some of these Alzheimer's diseases. And also in terms of aging, there is another gene called AGE1 in the C. elegans. When this gene is mutated, rather than living for 2 weeks normal condition in the lab, when AGE1, you can see, you can track the lifespan, not the life cycle. 2 weeks the normal wild type one lives. You found out a mutant called AGE1. It lives for almost 1 month or almost 2 months. So that means it is living longer. So what is this gene? How it can control the longevity compared to small worms dying in 2 weeks? How it can make them to live almost 2 months? They were studying all these genes, again comparative studies. They were able to find out that this AGE1 is nothing but a specific kinase family protein, phosphoinositidase 3 kinase. This protein is the one that controls stress resistance, longevity, and cytotoxicity also. When you look more into the pathway, how it controls, the PI3 kinase is here. This is the one AGE1 gene codes for in C. elegans, and it has a lot of crosstalk with several signaling pathways where they can conclude at the end what is the function of this protein interaction or signaling pathway is that either proliferation or pro-survival or apoptosis. They control a lot of the things where some certain cells are destined to die. When this pathway is compromised, the cells are not dying, they are living longer or they're surviving longer. So that's a way both aging and neurodegeneration you can study using C. elegans.
Sure. Thank you. So this is the end of the Q&A session because these are all the questions which we have got as of now. So
Okay.
Thank you very much, Dr. Nagagireesh, for your enlightening and an amazing session. And as we have been working a lot on gene and genome editing by CRISPR, Cas9, Cas9 nicases, but then obviously we are not into research, we are into trainings and workshops. So
Yes.
we have been doing a lot of workshops. And in fact, we have started doing workshops on CRISPR-Cas9 since May 2017. So that is what we have been doing. And at international level also, we have done many workshops on CRISPR-Cas9 and we have got a very good response also. So, thank you very much for your nice and, valuable time. And thank you very much. If you want, you can leave. However, I would just like to address the participants that the feedback link will be sent now, and you have time till tomorrow to fill up the feedback form, and then you can get the certificate. So the feedback link will be shared now. It is in the chat box, and you just click on it and just fill up the feedback. The link is available, or it is on only till 1 p.m. tomorrow India time. So after that, we will stop accepting the responses. So you have a lot of time to give the feedback. And thank you very much, Dr. Nagagireesh. And now, if you want, you can go ahead and leave the meeting. Thank you very much.
Okay, thank you, Asif. Thank you all the participants. Hope you have a good night, and thanks BDG, team BDG. Okay, take care you all. Bye.
Thanks.